Hello, my name is Chad Kinney, the Director of the Institute of Cannabis Research, hosted at Colorado State University, Pueblo. Thank you for your interest in our webinar series, and specifically the December edition of the series. Unfortunately, there was a small glitch in the recording of the webinar that resulted in the loss of the very beginning of the recording. However, it was only the introduction and the first slide to slide and a half that was lost. Therefore, almost all of the presentation was recorded, and I'm re-recording the introduction to our speaker now. We are pleased to have had Dr. Mahmoud ElSohly with us to deliver the webinar. Dr. ElSohly is the president and laboratory director of ElSohly Laboratories. He serves as a research professor in the National Center for Natural Products Research, the Research Institute of Pharmaceutical Sciences, and a professor of pharmaceutics in the School of Pharmacy at the University of Mississippi. Dr. ElSohly is the director of the Marijuana Project at the University of Mississippi, which is funded by the National Institute on Drug Abuse. Dr. ElSohly received a bachelor's degree in pharmacy and pharmaceutical chemistry, as well as a master's in pharmacy and pharmaceutical sciences from Cairo University. He earned a PhD in pharmacognosy from the University of Pittsburgh. He is board certified by the American Board of Forensic Medicine and the American College of Forensic Examiners. Dr. ElSohly holds more than 30 patents dealing with testing and detection of drugs of abuse, along with other patents dealing with biologically active natural products and compositions for the treatment of cancer. He has authored over 250 scholarly articles and delivered more than 200 presentations at scientific meetings related to drug discovery, analysis, and metabolism, and many of his articles deal with forensic issues of drugs of abuse. This is the second time that the ICR has had the pleasure of hosting Dr. ElSohly for a presentation. Dr. ElSohly delivered the opening plenary address at the ICR conference 2019. We are very grateful for Dr. ElSohly joining us to deliver this webinar. Thank you.
Yeah, an IDA-funded project that is focused on making materials available for research. And we just celebrated last year the 50th anniversary of the continuously funded project, which is the longest continually funded project in the history of NIH, which is we're something to be proud of. But the project focuses on making materials available for research, making genetic materials, making the actual products for experimentation, whether it's the cannabis plant material or the bulk quantities of extracts or some of the purified cannabinoids. But also part of the project is monitoring the potency of confiscated materials coming into the on the illicit market. And now, in order to meet those requirements, we have indoor growing facility to make it possible to make materials produced from through vegetative cuttings, and we also use that same grow room to produce some materials that special products that might be required for certain potencies or certain variety that we can collect from a small production site. Most of our production, however, is an outdoor facility, and outdoor growing is a large, about 12 acres, plot. But of course, we don't grow 12 acres every time. But every now and then we grow maybe 1.5 acres or so to meet the requirements of the national program. And all the materials that we produce go into the drug supply program, from which investigators can order materials from NIDA. This is the field, and I think that particular field here was 2014 when we produced different types of materials, all the three different chemovars that encompass everything that can exist on cannabis lies within those three chemovars. Now, at the end of the season, you harvest. These are the buds that are harvested at the end of the season, that really get processed and then put into the final product, which you can see on this one right here. The material, unlike what you find on the dispensaries, in the dispensaries you can get some buds here and there. You get some products that are not really processed, but in our product or our program, the material has to be manicured and processed in such a way that the final product that you see here is a coarse material that will lend itself to the manufacture of cigarettes, as you can see here. And that's the main product from the raw material, the cannabis itself, that goes into research programs, whether it's for human research or animal research. They use the cigarettes. However, the plant material itself, for some investigators interested in getting some material, let's say for vaporization, then they can either get the cigarettes and then take this out and put it in the vaporizer, or they can get the bulk material. Now, this material here is processed to the point where there are no seeds. Of course, if you are growing from a vegetative process, then you're not going to have seeds, but even with that, still you might have some seeds. And then stems or stalks and bigger particles or things like that are removed during the processing, so the plant material is nice and clean and it be easy and ready to go in the manufacture of the cigarettes. I might tell you that we don't necessarily all the time have material harvested and processed to give the exact potency that is needed for the program. So sometimes we do blend different materials together to come up with the required ones. Now, this material here serves as the basic for producing other products, such as the extracts and the purified cannabinoids. So in order to make the extract, we have traditionally and have really always been using organic solvent extraction with hexanes to make the extract. This is where we put the material into this stainless steel container, and then we do the extraction. Then we take the extract and evaporate it, put it through the evaporation process to remove the solvent, and you end up with the extract that then gets. Of course, we have to make sure that all the solvent residues have gone away, but we also do testing for solvent residue at the end. And that product that comes out at the end is a very thick tar, and we can only pour it when it's like around maybe 60 to 80 degrees C, and then you can put it in those buckets and it gets stored. This is the final product that comes out, and it remains in quarantine until such time as we do all the testing, whether it's cannabinoid testing or terpenes or aflatoxins, biological bioburden, heavy metals. All this testing has to be done on the extract at the end before we certify it and release it for distribution. This is every type of material that we prepare, the extracts that we have. We have two types of extracts. Actually, we have three now, and each one of those extracts represent one chemotype of the plant material. So we have a high THC extract, we have a high CBD extract, and we have the intermediate extract, which is an extract that contains roughly 50-50 mixture of CBD and THC, and that is coming from the intermediate plant material as defined by the THC and CBD content. In each one of those, we do have drug master file for the high THC material and a drug master file for the high CBD material. So if you are doing an investigation and you want to use one of those products, you don't really have to have in your IND application, you don't have to have a CMC section in that IND. Rather, we would give you authorization to reference our IND for those extracts, and that satisfies the requirements of the FDA. If the FDA needs to know exactly how things were done, they go back and they examine or review the IND for the particular product. The materials, when you order this material, they will get a certificate of analysis that outlines all the different types of testing that we do to make sure that the material is fit for use. So the COA for the cannabis extract, cannabinoids, terpenes, aflatoxins, heavy metals, the solvent residue, and so on. So that by doing that, you satisfy the needs or the requirements of the FDA. In order to go through take that extract, then the extract that was prepared, whichever extract that you start with, to make a purified cannabinoid, the very first step that we do in that process is distillation of the extract. As you can see, the crude extract is very dark in color, has a lot of chlorophylls in there, has a lot of hydrocarbons, so many different components in there. And by using that, what we call the thin film distillation unit, it really removes a lot of those things and does not subject the extract to high temperature for any length of time, so that you don't get much decomposition at all. So this is the first step in the preparation of purified cannabinoids from the crude extract. And then, of course, we go through different processes, whether it's chromatography or crystallization or whatever you have, method that you elect to do. We end up in the final product. We have, this is the THC. THC is always, you have this color associated with it, which comes from a little bit of oxidation of the THC to the dihydrocannabinol or cannabinol, which are very, have a strong color. But that doesn't mean that that material is not pure. It probably be 99.8% or so, but yet the color is not indicative. As opposed to CBD, like you have in here, where CBD is easy to crystallize. So you have, CBD as a crystalline material, and all this A, B, and C are crystals of CBD that are over 99.9% pure. And for the CBD, we did actually prepare some GMP CBD that we provided to NIDA and the FDA. That's the first material that was made GMP CBD for that purpose, to go into the clinical trial to determine the abuse potential of CBD, and that material we prepared here at the University of Mississippi. So we have not only this is the GMP, what you call the batch production record, that has all the information on that, which we submitted to the FDA. And we do have a drug master file for the GMP CBD. So if you have a need to use CBD from NIDA, a GMP CBD for a clinical trial, you can get that from NIDA. And then you also be able to give you a letter of authorization to reference the drug master file for the CBD, so you don't have to provide the CMC section. We also have now, we're working on one for the THC, GMP THC, and but that's not in the NIDA drug supply program yet. Now, if you look at cannabis and the history of the use of cannabis over the years, thousands of years, and you look at what on the market right now, medicines from cannabis, really, it's just, you can count on one hand. Marinol, which is a synthetic THC approved by the FDA back in 1980, and it has indications only for nausea and vomiting cancer patients, and for appetite stimulation of AIDS patients and also cancer patients. But I'm sure that a lot of physicians prescribe it off-label for some other indications, but in any case, just one product that has the THC. Then we have Sativex, which is actually a crude extract of both mixture of the CBD variety extract and THC variety extract, so that the final product, Sativex, is like a 50-50 mix between CBD extract and THC extract, so that the amount of CBD and THC is about the same per dose unit. Then Epidiolex, which has been approved only, I think was last year, it's about 98% plus CBD with a few other cannabinoids in there, and it's approved for the treatment of epilepsy, and is on the market now and has been changed. The regulatory status of Epidiolex changed from Schedule 5 to unscheduled, right now, just like CBD is. And then, of course, succinate, which is a synthetic cannabinoid that is also used, approved by the FDA about the same time as Marinol, and that is also for the nausea and vomiting with cancer patients. Now, if you look at the market today, you go to any drugstore or any supermarket or anywhere you go, you can see a lot of different products, different shapes and forms and so on. So the market has really opened very heavily, and people are selling so many different products and making some claims that are not substantiated by clinical trials or any research activities, but nonetheless, it's over there on the market. But none of those products has actually gone through the regulatory process, so they're not counted as drugs or medicines per se. But people say maybe dietary supplements, but FDA has not recognized those things as dietary supplements. But they're putting it out there for refractory childhood epilepsy, which is the indication for which Epidiolex was approved. Now, so these are the things that we have contributed to making and getting those materials out for research. Now we have been focusing our activities on not on the dietary supplement side or on the just making materials available on the market, but rather on developing pharmaceuticals from that are cannabis-based pharmaceuticals. And I'm going to share with you, two products that we have been working on for the last few years, and they are right now have been licensed to a company on the West Coast, and hopefully that it will make it to the market. But it's a lot of work and a lot of activities that have to go on for that to happen. Anyway, so we start out with the THC products. Some of you might recall my previous work years ago on suppositories, making a THC prodrug suppository. And that has really been licensed to two different companies. Unfortunately, they did not really carry it through the development process not legitimately, but diligently, I should say. And therefore, it never made it. So just a few years ago, I decided that and the patent on that particular product had expired or about to expire. So there was no future or no hope for getting it on the market. So I developed some new set of compounds and derivatives that revived, that patent, and now we have a product. And I started out with making just the hemisuccinate and hemiglutarate in the beginning, in the old program. But then now we shifted. Now we make this compound right here, delta-9 THC valine hemisuccinate, which you take delta-9 molecule, and you attach that in an ester form with an amino acid valine, and then to the nitrogen of the amino acid, you make the amide with the succinic acid or succinic anhydride, and that gives a very good stability to the chemistry of the compound, but also continues to make it bioavailable through routes other than the oral route. And that's what we have been trying to develop this compound. So I'm going to share with you some of the pharmaceutical formulation that we have developed, starting with this particular product right here. Now, the first one is a suppository again. And, in this publication that I have cited down here, this shows the old suppository, which was made with the THC hemisuccinate, which had a very good efficacy, very good profile, but was not, very stable over a long period of time. Shelf life was not too good. Had to be done in a lipophilic, base, and the bioavailability was still, limited because it hydrolyzes fairly quickly. Now, delta-9 THC itself, as you know or might not know, does not get absorbed from the rectal mucosa or any mucosa for that matter. So anyone that tells you that we make THC, we take THC and put in a suppository and it works, it doesn't work. It does not get absorbed. It could be just a placebo effect. So you really have to derivatize the THC structure to be able to make it to go through the mucous membranes. This graph right here, and I don't know whether you can see maybe you have a bigger screen you can see, but you can see this here with the blue one is the delta 9 THC content, and you can see here up to 40 something nanograms per mil, which is a tremendous amount. This is out of a 7.9 milligram per animal, and this is done in rats, in cannulized rats. And then the red one is the 11-hydroxy, which is much lower than the THC, and that indicates the bypassing of the first pass effect, because if it's an oral preparation, then you see about equal amounts of THC and 11-hydroxy THC. And then the green one reflects the carboxylic acid metabolite. So you can see very, very good bioavailability from that formulation, and it continues. If you're starting out with time 0 and it goes here at I think this is like 15 hours, you still have like 10 or 15 nanograms per mil, and it continues almost. So which means that this particular product would be administered just once a day, as opposed to Marinol, which you would have to give that every 6 hours or every 4 hours, depending on the condition, to be able to sustain enough blood levels to get efficacy. In this case here, you only need one suppository a day, and which is another advantage of this particular product. Just taking a lower dose of that, I mean half the dose, you still have above 20 nanograms per mil. This is about 18, 20 nanograms per mil of the THC, and then about 4 nanograms per mil of the 11-hydroxy, much lower level. Yeah, again showing that the absorption through the mucosal membrane goes directly to the systemic circulation before going through the liver to be metabolized. And then again, the green is the carboxylic acid, the final phase 2 metabolite of THC. So you can see from these two slides that the bioavailability of THC from the suppository in using the valine hemisuccinate derivative is tremendous. And I can, if this translates directly to humans, I would say that the dose in humans will probably be no more than 1 or 2 milligrams, which is a very, very small dose, and it gives you enough blood levels to provide the efficacy that is needed from THC. Another formulation that we developed with this particular compound is a transmucosal matrix patch or delivery system, whereby we made a disc, a small disc, like you see here on this, applied above the gum line, and then it has a bioadhesive that will allow it to adhere to the mucosa, and then it has a backing. The final product actually will have a backing on the back here to prevent it from going here and there, and then the slow release of the product through the mucosal system to get into the general circulation. We tried that product with 2 different compounds, the THC hemiglutarate, the one that these are the 2 compounds that I showed you the structures in the beginning, and you can see that from either one of them, it works the same way. Now and so what we decided to go with the Val-HS simply because of the shelf life stability. This Val-HS is a lot more stable than the hemiglutarate, and therefore we continued with the Val-HS. The third formulation that we worked on using the same product is the eye drops for ocular delivery. Everybody knows that marijuana was since 1971 was found to reduce the intraocular pressure, and the reason for that is because of the THC and the interaction with the CB1 and CB2 receptors in the eye. And but knowing that the eye is not a very easy organ to work with, it has a lot of barriers, and therefore Delta 9 THC was tried in the eye years ago, years and years. They're trying to get it, but it doesn't really go through. And we made this product here. That product is capable of penetrating, getting into the inner compartments of the eye, and then there it converts, it breaks off to deliver the THC. So the derivatization of the THC and making the VAL-HS derivative is really just a way to allow the THC to get in the form of the prodrug, and then once it gets there, it breaks off, releasing the THC at the site of action and producing the effect. This this is the ocular distribution of delta 9 THC through topical application. This is just THC, not derivatized. And you can see here from this, there is no absorption, no going into any of the inner compartments of the eye. It only stays on the cornea, on the top. That's where the only place that you can see THC, but then inside the eye, no THC. So as opposed to if you prepare the derivative, like in this case here, the THC hemiglutarate, you can see those compartments. Those are highlighted here. You can see that you do have some levels getting into the eye, but it does not reach into, does not go into the vitreous humor or reach the retinal choroid. But nonetheless, the cornea, the aqueous humor, and the cellular body, iris cellular body, it goes in there, and these are really the areas of the eye where the delivery of THC and the interaction with the CV1 receptor there, what just causes the lowering the intraocular pressure. Here's our compound, the THC VAL-HS, the valine hemisuccinate. You can see in those three compartments here, again, you have much higher levels than the hemiglutarate. And then what we have been trying to do, different types of formulations. And then here is what the ocular film. And the ocular film turns out to be the most efficient way of getting the material to go through, even all the way to the retina choroid. So we in doing this, we knew that it's a matter of finding the right formulation to get the material to go. We know that it goes into the eye, but where does it reach? How far does it go? We didn't really know. We know that it does not from the regular formulation, just a regular emulsion form, emulsified THC VAL-HS. It doesn't reach all the way back, but we can put it in a film, which give it more exposure and allows it to go all the way to the retina choroid. Now, this is here the ocular distribution of this material from hydroxypropyl beta cyclodextrin solution versus an emulsion in cremophore RH, and along with other components that we're adding in there versus the ocular film. Again, you can see that the ocular film is has higher levels than the other formulations, and it reaches the retina choroid. This is again comparing the 1 hour versus the 3 hour between the different formulations here, and showing that the ocular film is really the way to go, that gives you the highest exposure. But we have, since that time, we have also worked with some other formulation I'll show you in a minute that allow the THC VAL-HS activity to continue for several hours. Okay, now we just talked about the fact that the activity of of delta 9 THC in the eye has something to do with the binding with the CB1 and CB2 receptors. And if you look at this compound, the THC VAL-HS, and how it interacts with both CB1 and CB2 receptors, really the binding is really not, is very low compared to THC itself, indicating that when the compound goes into the eye, it has to break off to release the THC, which then interact with the CB1, CB2 receptors to produce the activity that we're looking for. But the compound itself, it's a prodrug, so it doesn't really show its activity until it breaks off releasing the drug. We developed a glaucoma animal model that we use to evaluate the activity of this particular compound in the eye. And this is the alpha chymotrypsin model that will allow the intraocular pressure to go up. And once it gets stabilized, then we can use those animals to evaluate the effect of the drug of the formulation on the intraocular pressure and how effective the compound might be. We looked at different materials here. These are different formulations and different compounds. Here is delta-9 THC itself. We tried delta-9 THC itself, different concentrations and different emulsion. Tocresol is one of those things. We can get some reduction in the intraocular pressure when you put delta-9 THC in emulsion form. But the activity is very transient, just 30 minutes, 60 minutes, by 90 minutes is gone. So, and then, but it does lower the intraocular pressure, but it's not stable enough. Then you look at these are different formulations of the THC val-HS versus THC itself. Here is the THC in the same formulation, right here at 0.8%. This one right here is up here, and then is again, like we saw before, it comes quickly and then disappears. Then but you look at that one with the green, which is delta-9 THC, 0.6%, so it's a lower concentration than THC. Look at how deep the curve is, and the lowering is maximized at 2 hours, but it stays till here about 240. That is like 4 hours. We know that this is good, but it's really not good enough, because we need the drops to be maybe no more than 3 times a day. So it needs to have to stay there, have a good lowering of the intraocular pressure for at least 8 hours. So we're continuing to work on this, and we have started working with some other formulations. If you look at this here, for the same formulation that we had, this is timolol, and timolol has a longer duration of action. And this is our compound right here, as we saw in the previous slide. And therefore, what we need to do, we need to shift the activity to have it to stay longer. And we have been able, to do that with modifying and working on some other formulation, like solid lipid nanoparticles or nano emulsions. Here is a solid lipid nanoparticles. You can see now this is the contralateral eye, where we put the same compound in the solid lipid nanoparticles right here. In the contralateral eye, we didn't give, we just gave the vehicle, but then in the other eye, we gave the compound. You can see the lowering of the intraocular pressure here, and continues for, this is, let's see, about 480 hours. So that is 8 hours right there. And you can still see some reduction in the intraocular pressure. So in order to conclude this work that we have been doing with the THC val-HS, basically we can tell here that the hydrophilic derivatization increases the ocular tissue penetration of delta-9 THC, and the solid lipid nanoparticles and matrix films can further improve the ocular penetration and retention of the compound, and of course the duration of activity to make it maybe 2 or 3 times a day rather than once every 4 hours. The other compound that we wanted to do that we've been working with and making some derivatives of is CBD. CBD, of course, now is a very well sought compound for a variety of reasons. It has a lot of good biological activities, but then again, the oral bioavailability of that compound is not very good. And also, for example, penetration into the eye for CBD is not good at all, just like THC. So you can make formulations, but then it doesn't help. Anyway, so we took the same concept that we did with the THC and applied that to the CBD. Here's a CBD, molecule right there. And then you have, of course, you have two hydroxy groups here. So when you derivatize that, you can derivatize one or the other. I mean, the other would be, these two hydroxy groups are equivalent. It's a symmetric molecule, and therefore you either make one or you make both. The di derivative is not really that same. We have looked at the mono, looked at the di, looked at putting the mono val HS on one hydroxy and just HS on the hemisuccinate on the other. And we found out that the compound that has the monovalene mono HS, which is this one right here, is the best compound to work with. And as it turned out, the CBD monoval mono HS is not really, although we started out, thinking that we are going to be preparing a prodrug, it turned out that the monovalene mono HS of CBD does not act as a typical prodrug, so it would not pass as a prodrug. It would pass as a new chemical entity simply because it does not behave in the body as a prodrug. For example, if you take the delta 9 THC hemisuccinate, valine HS, the compound we just talked about before, and you put that into the animals and you go into the blood and you look, you would expect to see the THC because it's supposed to break off, and you do see THC. On the other hand, if you take the CBD monoval mono HS and you put that in the animals, I was not able to see the CBD in the blood. I only see the CBD val HS. When it goes into the tissues and starts getting distributed and so on, then it starts breaking. So the breakdown of the CBD monoval mono HS to release CBD is a slow process, but yes, it does happen, but it's a slow process. So basically, once we figured that out and we found that out, we realized that the only way that we can have a good drug out of that compound is if the compound on its own is active, unlike the typical prodrug where it has to break off to release the drug before you can see activity. In this case here, we looked at this, looked at the CBD val HS versus morphine in the cisplatin-induced neuropathy. Cisplatin-induced neuropathy. Here is, I'm just trying to make it simple for us to look at. This is the vehicle. This is a low dose of morphine, 0.1 milligram per kilogram. This is 2 milligrams per kilogram of morphine, and you can see that it does have good activity. This is the normal here. This is increasing the mean withdrawal threshold from the pressure put into the paw of the animals. Then here is here are some different doses of the CBD val HS. You can see this is a small dose here. I believe this is 1, 2, 3, and 4 milligram per kilogram. And you can see that at the 4 milligram per kilogram, you have equal activity as 2 milligrams per kilogram of morphine. That gives you very good indication that the compound does have a good analgesic activity, at least in the cisplatin-induced neuropathy. Then we try to do some work to see about the potentiation of the effect of morphine using the CBD monoval mono HS. We can see that at 2 milligram per kilogram, which was here by itself, here is with a 0.1 milligram of morphine, you can see have activity similar or almost equivalent to 2 milligrams per kilogram of morphine, or 4 milligram per kilogram of CBD val HS itself. But then here at the 4, you really don't get any more activity than what you get with the CBD val HS on its own. So we figured that for CBD val HS, you don't really need the morphine with it because it does produce activity similar to an effective dose of morphine. Then we tried that CBD VALHS in another analgesia model, which is the abdominal writhing, and you can see that oxycodone gives a very good activity by itself, and then when you combine it with the CBD VALHS, you can see that it just totally eliminates the abdominal writhing. That indicates that there is a good positive interaction between CBD VALHS and oxycodone in the abdominal writhing model of analgesia. So we have done, I'm just in the interest of time, I didn't really put a lot of different slides along those lines, but just suffice to tell you that we did some work with the CBD by itself. We did some work with the CBD, with the morphine, with oxycodone, and so on. And then this is just summarizing all this work that we did along those lines in that one slide here. CBD does block morphine reward, and then CBD VALHS also blocks oxycodone reward or morphine reward. But neither CBD nor CBD VALHS show rewarding activity or aversive activity either. And then CBD VALHS with oxycodone retains analgesic effect on a clinically relevant pain assay. And then CBD VALHS produces analgesia when given alone across pain models. And CBD VALHS alone provides better pain coverage than plain CBD, underivatized CBD. And then high dose of CBD VALHS alone provides comparable activity as we show to to morphine. And when combined with a minimum dose of morphine, provides comparable pain coverage to high dose of morphine alone. Meaning that the CBD VALHS appears to be a very good compound to use for analgesia without having the abuse liability that's associated with the opiates. Now, this is one final, one or two slides final to bring you up to speed on some of the things that we have been doing with the CBD rich extract that we have, that we talked about earlier in the presentation, that we have, prepared, because we here in the state of Mississippi, they mandated that we make some CBD oil available for clinical trials in Mississippi. So we took it upon ourselves. They didn't give it, although they required that, but they didn't really give any funds to do it. So we made the extract. We have the GMP material made. We have the drug master file on the extract, and we have everything in place. And so we had to work with the medical center in Jackson to be able to make it and deliver it to them to do the clinical trial. So what we decided to do is actually provide the extract in these bottles right here, because that was the most expeditious way. So you don't have to do a lot of stability studies and so on at the and consider it like a drug product per se. But anyway, they would take this, and we gave them the formulation, how to dissolve it in the sesame seed oil and all of this, and then start administering that. They're giving it at starting out with 100 milligram a day, and then move to 200 to 300 milligrams a day to the children with the epilepsy. And the trial has been going on now for over a year, and we were able to provide them with this material here. We acquired the CBD rich extract through the NIDA drug supply program, but then we made the material here and shipped it to Jackson, and they were able to deliver that. The trial is going very well. There is no no adverse events really to speak of. And the children, the parents of the children have been very delighted with the results of the use of that product. I think with this, I will conclude by thanking my co-workers that work with me here at the Coe Waller complex. And we have a lot of good people helping me with all of this work. And of course, the National Institute on Drug Abuse for funding the majority of the work that we do here. Of course, the development of these, the CBD VALHS and the THC VALHS are outside of the NIDA drug supply program. But nonetheless, they made materials available to be able to carry out those types of activities. And I thank you for listening, and I hope I can answer any questions you might have.
Thank you, Dr. ElSohly. We do have some questions that have come in. So the first question is the GMP CBD controlled and greater than 0.3% THC?
The which one?
The GMP CBD.
No, the GMP CBD now is, so long as it has less than 0.3% THC, is not controlled. It's outside of the Controlled Substances Act. And my opinion is, and don't hold me to that, my opinion is whether that CBD is natural or synthetic, doesn't matter. So long as it has less than 0.3% THC, is not controlled. But there is still some thinking that maybe DEA would say the synthetic CBD is, even though with less than 0.3% THC, is still controlled, but I don't think so. I don't think they can really justify that.
So that's a great segue. I think you're speaking to this right now, but the question came in of using the GMP CBD still requires a DEA C1 license, correct?
No. No, it'd be just like GMP aspirin or GMP Tylenol or GMP any other drug. You just have, I have actually, well, I was going to say I made that, but I haven't. I made it before, yeah, that before the CBD came out of the Controlled Substance Act. I don't think it would require a DEA 222.
Okay, thank you. The next question is, the NIDA website lists that the highest THC flower available is greater than 10%. What exactly is the highest THC concentration in your flower?
Well, we have actually, we just remember that that the program from day one was really intended to make materials available for research and make material available that can actually go into clinical trials using the commonly used method of ingestion, which is smoking. So that's why the program was designed to make cigarettes. And you can show in the slide that I had in the beginning there, you saw the cans of cigarettes there with the 300 cigarettes in each one of those cans. And those cigarettes are about somewhere between 0.8 to 1 gram on weight. And these are standardized NIDA standardized cigarettes. And therefore anybody that is to do a clinical trial with the NIDA cigarettes, in order to make it a placebo-controlled double-blind study, neither the subject nor the investigators need to know which is which. They all made and they provided, and therefore they had to require the subject to smoke the entire cigarette, because that's the only way you can really tell that the only difference between this product and that product is the amount of THC in there. So we had prepared things from placebo to 1%, 2%, 3%, 4% for many, many years. And the maximum was like 4% until 2000, maybe 2005 or 2006, when we had Dr. Abrams from the University of California, San Francisco, said, I need to do a study, and I need to go to do my study to go from the placebo, 2%, 4%, 8%. So I need 8% cigarettes. Well, we didn't have 8% cigarettes because the maximum we had back then was like 4%. So I said, okay, well, we don't have that, but we can make a small batch, hand-rolled cigarettes, exactly the same size, same everything, but with 8% material. So we made that. With Dr. Abrams got the material, started the clinical trial, and the most experienced marijuana subject, smoking experienced subject, could not finish the 8% cigarette, could not tolerate it. So he called me and said, Mahmoud, you know, my most experienced subject can't tolerate this thing. You cannot finish it. So can we make the highest to be 6%? So we went back and the reason we only made a small batch of the 8% is the higher the amount of THC in the plant material, the more sticky it is. And then when you put it into the high speed, high volume cigarette making machine, it jams the machine. You can't do it. So the high potency, you have to do it by hand. But the 6% was no problem. So in that year, we manufactured 60,000 6% cigarettes and put those in the program, in the drug supply program. And we made the 6%, and so he took the 6%, finished the trial. Of course, it was double blind and everything, and the questionnaire. Found out at the end that most subjects actually preferred the 4%. Some subjects preferred the 2%, some subjects preferred the 6%, and some subjects preferred the placebo. But more than 70% or 60, 70% of the subjects preferred the 4%. That's the one that gave them the most benefit, and whatever it is they were looking for, the 4% was it. So the program is designed really to prepare things that the subject can tolerate. We're not trying to make things very high in THC, because to do a study where you let the subject use the material ad libitum is not a controlled study.
Okay, thank you.
Because you don't know how much they use. But to come back to answer your question, we have prepared material as high as 20%, but not in large amounts.
I see.
Just a small batch that was required by one investigator, and we have another one now in California that's needing that. So we make a small amounts of the high potency, but not on a large scale.
Okay. This next question is two parts. Do you have any Delta 8 THC products that are available for human testing, or any products with isolated minor cannabinoids?
We have, but we're not having it in the. I think the drug supply program does have some material, but not in bulk.
Okay. And then how do the terpene profiles of your hexane extracts compare to those of the plant material itself?
Obviously, in the terpene profile of the extract, because the extract you extract with a solvent and you evaporate the solvent, some of the monoterpenes are diminished in the extract than in the plant material itself. So you lose some of the terpenes in the distillation process to remove the solvent and to do the decarboxylation of the extract.
Okay. This next question says, one of the first slides showed cannabis cigarettes. Do you feel that mode of consumption will hit the market as an approved method before other oral methods of consumption?
My personal opinion is that the smoking is not a viable pharmaceutical delivery system. That's my view. Now, having said that, it is also important to remember that a lot of companies now are making vaporizers that do not ignite the plant material. They just heat the plant material to a certain degree to vaporize the THC or the CBD or whatever the cannabinoids and the terpenes, but not burn the plant material. So and those I have seen at least one of those devices that they're really very precise in terms of delivering how much they would deliver and they would use. So they're very efficient, very effective. So there might be an approval of a cannabis plant material in a vaporizer, but not in the cigarettes like we use tobacco and so on. I don't think that's going to happen, but I could be proven wrong. Now I have to tell you also that the way of smoking of the material, you don't really know how much exactly you are getting. But with the vaporizers, there's a lot more control over the dosing. than it is with the cigarette smoke.
And I think this is going to be the last question that we're able to address today. And it says, does the cannabis powder supplied by your program come with a warning that it should be irradiated to destroy mold spores that may be present?
Well, actually, we have, Nicole, we have started from the 2014 production, and we produced in 2017 or just this last year, 2019. All the material that we have has already been irradiated. We're actually preparing a manuscript right now. Maybe I'll send that to Dr. Kenny. Maybe I'll send that article to your journal to publish there. And we have taken the plant material, all the stock that we have. I think we had like over 1,000 kilos of plant material, different potencies, different chemical composition, different profiles, and we have irradiated each and every container that we have. And we have done a lot of work with the irradiation. This is gamma irradiation, by the way. And we looked at the cannabinoid content before and after. We looked at the terpene content before and after. We looked at the moisture content before and after. And of course, we looked at the bioburden, both the yeast and mold content and total aerobic organisms. And of course, in the beginning, you have high levels because that's materials grown outdoors. So you would expect that the normal bacterial flora in the atmosphere would be there. And then after irradiation, it's basically sterile product. So we are doing this already, so we don't have to put the warning.
Thank you so much for your presentation, Dr. ElSohly, and for answering all of these questions. We really appreciate you joining us today and for all of the efforts that you've helped the ICR with. For attendees, please note that the full webinar will be available on the ICR website in just a few days. The next webinar in our webinar series will be given by George Hodgin, and his topic will be access to federally compliant cannabis for research, a long and winding road. This concludes today's webinar. Thank you, everyone, and have a great day.
Thank you, Nicole, and thank you, Dr. Kenny, for the invitation. It's been a pleasure being with you today. Thank you.
Thank you.
Thank you as well.